All right, good morning. Dear student, the same thing. I need you to take a piece of paper and a pencil. And what we are going to study today is another type of metabolism or another type of chemical reaction called photosynthesis. And who are the organisms that are in those type of photosynthesis? Those are the organisms that they have as organelle chloroplasts. Of course, this is a plant, all right? So let's see what we give the plant. We give them water, all right? This is what they got. They capture the sunlight. It's so amazing. All what they did is to capture the sunlight, which is the source of energy. That's the universal energy, is the sunlight. They capture the sunlight, and in the atmosphere, we have carbon dioxide. So they capture this carbon dioxide and they use the water to produce organic molecule. That we are eating. So imagine you are eating a piece of salad when you are hungry. So you are eating actually sugar that has been made by this salad. And how the salad made this sugar, this organic molecule that you are eating, is by converting or fixing, because you know carbohydrate is a carbohydrate, is a carbon molecule. The only source of carbon that I have seen here in this plant that you are given is a CO2. It's amazing. I want you to see how that's possible. They capture the CO2, transform it into sugar. And not only that, they release oxygen. This release came from water. So not only they are going to capture the CO2 to produce the carbon, but they are going to hydrolyze the water to produce oxygen. So the general equation, and I want you to take a piece of paper. I want you to write it down. This is your plant, okay? So let's do the same thing. I'm going to take a piece of paper. So we're going to have, we are plant now. The cell is a plant. What we give to the plant? Water, right? CO2, and they produce an organic molecule. So we are going to make it very simple, this organic molecule that they are producing, okay? We are going to simplify it as we simplified also for cellular respiration. We are going to make it like it's a molecule of sugar. The basic source of energy for the cell that they use, to catalyze to produce ATP. C6H12O6, sorry, plus oxygen. This is the plant. So I'm going to balance it. This is the plant. And what else? What am I going to make this convert into this solar light? So in another word, the plants are able to convert solar light, use it with the water and CO2 to produce an organic molecule, to produce the fuel of life. The fuel of life. This is what we are, remember, animal, this is what they are using. C6H12O6 plus oxygen. So it's almost the reverse of cellular respiration. The oil that I put in red, that's a cellular respiration. This is what the cell do to harvest energy. So we are going to use water, carbon dioxide, sunlight. that are the reactant for photosynthesis, that are the reactant for this metabolism, this chemical reaction. Carbon, this CO2 is just one carbon molecule. To produce C6H12 is big molecule. But remind you this is anabolic or catabolic reactions. When I build small to big, this is an anabolic reaction. Is it exergonic or endergonic? Where it uses the light, it uses energy from the sunlight, it's an endergonic reaction. Therefore, the DG, remember the free energy, it's a positive. Who done those? Any organism that have photosynthesis, plant, algae, and some cyanobacteria. Plant, algae, some cyanobacteria, and some purple sulfurous bacteria. But this purple sulfurous bacteria instead they produce oxygen, they produce sulfur because they are not using CO2. They are not using water. Sorry, because the source of oxygen here is coming from splitting the water. They are using H2S. All right. So I go back again to the chemical reactions. And they really want you, this is an R over here, I want you to write it down. Light, sunlight plus CCO2 plus 6H2O, giving me C6H12O6 6O2. This is also a redox reactions where the water is oxidized and the CO2 is reduced. Remember, reduce when it becomes big, oxidize when it becomes smaller. And this to be formed, you need a lot of electron to be formed. It's going to, yes, you have 12 protons and increase on a number of electrons too. So let's see where this reactions, as I said, this photosynthesis happen where? In the leaf, right? Where exactly in the leaf? In what we call in a specialized tissue that we call mesophyll. Where exactly in the mesophyll? It's in the chloroplast. The mesophyll, if you remember, we have the cell wall, right? And we have some stomata, which are pores in the epidermis, right? And there in the stoma can be closed and formed to let the CO2 come in and to let the oxygen, when it's formed, came out. Inside those mesophyll, we have what we call an organelle that we call chloroplast. If you remember our lecture about the plant cells, the chloroplasts, they have an envelope also because it's a double bilayer of phospholipid. It's a double membrane, so it is an envelope. And of course, the one that they are in contact of the cytoplasm or the cytosol of the plant, we call it an outer membrane. The one that is inside and in contact with inside of the chloroplast, we call it the stroma. It's aqueous. And we call it the inner membrane. We have, of course, between them the intermembrane space, but this is the most important. What we have inside of the stroma, inside of the chloroplast, we have what we call thylakoid. And more than one thylakoid, we call it granum. Each one of those green stuff inside, it's called thylakoid. And more than one, we call it granum. They are stuck of membranous. And you know what they have inside? we have a thylakoid lumen or interspace of the thylakoid. We will see that sooner. So this is a granum. This is a granum. This is more than one thylakoid inside of the chloroplast. This is a granum, right? And this green line over here, that's the thylakoid membrane. Thylakoid membrane. Thylakoid membrane. The dark green that you are seeing all around. All around. So the thylakoid have a membrane, and inside of the membrane, it's the thylakoid space or the thylakoid lumen. So let me go back now. If you remember the energy, to have the energy come in, we have really to keep the hydrolytic system. I mean, we have to have water. If this is if they are not raining, for example, and the lake is completely no water on it, where it will be that system, it will be the delta G will be 0. It's a system that is in equilibrium. It is 0. So for the photosynthesis, what is the source of electron? The electron came actually from splitting the water. This is the source of electron. And who is the final acceptor of electron is the sugar. Because I told you to make this big molecule, it's an anabolic reactions, but those electrons came from splitting. We cannot split CO2. We can't split CO2. But the water, we can hydrolyze it. We can split it. And by splitting the water to oxygen, we are releasing electrons and proton. And where those electrons and proton are going to end up on the molecule of sugar, on the organic molecule. On here. The electrons, to reduce this, to making it big, they have to accept, they are the final acceptor of electrons. This electron came from splitting the water. And therefore release oxygen. And by the way, I mean, water need 12 molecules, water. The plants need 12 molecules that we realize. For each photosynthesis reactions, we consume 12 molecule of water, and we produce 6. In the end, we produce 6 H2O. But here, and this is 12. Here I'm making it, we use 12, and we produce 6. And to simplify it, 12 minus 6, because we are getting 6 and we consume 12, we are going to make just the net consummation. This is why we use only 6. But maybe in your book you are going to see, oh, the photosynthesis use actually 12 molecules of water. It produces 6 molecules of water. So it's not only that the plant consume water, but also produce water. But they consume more what they produce. So we are going to talk only about net consummation here. Okay? So, you know, the sunlight emit energy. The energy is is just an assemblage of photons. Is composed of particles that we call photon. Right? Photon. So let's see that this is the light. This is the photon. When they excite some compartments that they exist actually in the plant, where exactly in the plant? In the mesophyll. Where exactly in the mesophyll? In the chloroplast. Where exactly in the chloroplast? In the thylakoid. Where exactly in the thylakoid? In the thylakoid membrane. On the membrane of the thylakoid, we have some what we a photosystem, right? When the light stimulates those two photosystems, we have two that we know, photosystem 2 and 1. I will come back to this. The electrons came from a state that they are really, at rest, to an excited state. Go up. It got excited. You have to get rid of this electron that got on them. So they have to give it to another photosystem that is more electronegative than this one, and they are not going to give it to them directly. You have to go through an electron system transporter over here, very short one, and he end up in the photosystem 1. All right, but here is one thing that I want you to understand. When those photosystems in the thylakoid membrane, imagine them, they are just a molecule, proteins that they are embedded in the thylakoid membrane. When the sunlight excited the photosystem 1, you think that the photosystem 2, because he's less electronegative, he has been discovered after photosystem 1, so this is why we call it photosystem 2, but he is less electronegative than photosystem 1. So he is actually the first one that he is very sensitive to accept electron coming from splitting the water. He has very strong sensitivity to accept electron coming from the water. Not this one. This one is more electronegative than photosystem 2. He accepts electron coming from the photosystem 2. But he is not going to take it directly. He is very smoothly through an ETC, through an ETC, electro system, through an ETC, electrotransporter system chain. All right. So they are going to accept, but when the light, those photons are exciting photosystem 2, the photosystem 1 is not high. The light is covering all the plants. Okay. So they are going to excite it also photosystem 1. The electrons that they are in photosystem 1 go also from a state that they are less to higher energy. All right. So they got oxidized over here because they are going to lose electron, and they are oxidized over here to cover this oxidation, this loss of electrons. With this one will accept electron, from where? From splitting the water. And this one will accept electrons coming from the photosystem 1 through the ETC. I come back to this. You are going to understand it. So first of all, let's look at the two stages for photosynthesis. First of all, this is what we call dark reactions. And we have another one that we call late dependent reactions. The dark reaction does not mean that it's happened in the dark or in the night. It's happened that doesn't necessitate the sunlight to happen. But it's very dependent on what's happened there in the light reaction, the reactions that's depending on the sunlight. During these dark reactions, again, dark reaction does not mean it's happened during the night, okay? The CO2 is the one, this is the only objective for this dark reaction is to fix this atmospheric CO2. So the sugar, because the only source for carbon is coming from dioxide of carbon. I come back to this slide. So first of all, let's look at the nature of light. So light, it's an electron, is in the form of electromagnetic radiation, right? This radiation exists at different wavelengths. Wavelength is the single wave, the distance between two constitutive points at similar position. They have the same crest and the same trough along with the wave. So this is a wavelength. So therefore, all forms of electromagnetic energy travel at the same speed, same wavelengths. All right? Look at the wavelength for the blue over here. Look at the wavelength for the purple. They all travel in the same. Look at the wavelength for the red. They're different. Right? Their crests and the trough are different. Look at the crest for the red, the trough. Look at the crest for the purple. You know, it's different. So all the electromagnetic energy travel as the same wavelength. So the sunlight emits energy in the form of electromagnetic radiation. We have some radiation that we can see, they are visible to the eye, you know, visible radiation. And there are some that we cannot, they are we cannot see, right? And the light, I said, is always composed of massless particles that we call photons. One thing that is very important that I want you to understand, that all the color of the visible light do not carry the same amount of energy. Longer is the wavelength, less energy is on it. Shorter is the wavelength, more energy is on it. This is inversible, right? So now that we know that, so this is longer wavelength, less energy. Shorter wavelength, more energy. And the visible light go from blue, violet blue to reddish. Okay, that's our visible light. They are one researcher. His name, he's a British guy, that his name is Angelman. He was very feminist guy, but I'm not going to talk to you about how feminist he was. He was always for women in the sense, for vote, but anyway. So I'm going to talk to you about this smart experiment he done. You want to know which structures are responsible to absorb those wavelengths, those electromagnetic energy from the sunlight. He done something very, very smart. Take a plant, an algae, filamentous algae, right? Filamentous green algae. He take it and he grow bacteria. It's very funny. Those bacteria can grow only on presence of the oxygen. They are what we call aerobic bacteria. Follow me. They are aerobic bacteria. I hope that you got it. It's beautiful what he done. So he take this bacteria, make them grow in the top of the filamentous algae, and he is going to is going to submit those filamentous algae to different wavelengths of the light. Because this is aerobic bacteria. He put them in an environment, there are no oxygen. He's in an incubator, no oxygen. All what they have is different wavelengths of the light, filamentous algae, no oxygen. Bacteria to grow, they need oxygen. And he is going to use different wavelengths. So when he saw that when he put those algae in the blue light, blue purple light, like you can see here, what you can see, lots of those dark dots are bacteria, a lot of bacteria growing. That's mean. Where did they get the oxygen? I didn't have oxygen in my media where I put them in. I got it from the algae. So the green, the blue and purple is absorbed by the filamentous algae because my algae is doing photosynthesis, producing oxygen. Oxygen is one of the products of the photosynthesis. When he put them in the green and yellow light, bacteria are not growing, very less. Okay, because there are no photosynthesis happening here. When I put them in the red, again, I see a lot of bacteria, those black dots over here. Wonderful. So therefore, he came up by saying that the plant absorb in the purple blue light and the red light. And the pigments that they are in the plant responsible, because I can isolate them actually. I can isolate them. If we were in the lab, we will do it. But we will use some pigments. All what we do is browing the leaf of the plant until we got those green liquid coming out, and we are going to test it. Okay, so those plants are made, those pigments, they are three. He realized that we have three essential pigments. We have chlorophyll A, chlorophyll B, and beta carotene, and other hydrophobic organic pigments. This is what I found in my thylakoid membrane. Chlorophyll A and B, they are almost identical. Look at them. They are almost the same. And I realized that those chlorophyll absorb, A and B, absorb in the blue, purple. And the chlorophyll B, if you see it, it absorbs less in the red. It's always A that is in the peak. It's always A that's absorbed also in the same peak than in the purple and blue. So therefore, the major chlorophyll pigments are responsible of absorbing the sunlight is the chlorophyll A, is the most important pigment in the photosynthesis. I don't want you to retain this picture. Look at how many rings he has, and it's very complicated. The chlorophyll A is the most important pigment that absorb in the purple, blue, and red. The accessory pigments that they are going to play a role also. I'm not saying they are not. The most important one in the accessory pigments are going to be chlorophyll B and also carotene that absorb orange and yellow, and xanthophyll, which is yellow. Let's look at, I'm going to come back again to this, okay? Because here's the thing. Those are, you see those photosystems that I told you that exist in those membrane, photosystem 1 and 2. Here is the anatomy of those photosystems. They have a lot, they have what we call reactant center. and they have a lot of accessory pigments. And this reactant center, this is what they have, this pair of chlorophyll A. See those two inside over here? Those are there of chlorophyll. So the photosystem have inside what we call a reactant center, and between the reactant center I have a pair of chlorophyll A, and all those they are accessory pigments, accessory pigments, chlorophyll A, B. Chlorophyll B, xanthophyll, accessory hydrophobic pigments, all right? But the most important, those photosystems have those pair of chlorophyll A, because I told you, Engman discovered that chlorophyll A is the most important chlorophyll or pigment responsible for the photosynthesis to happen. The photosystem 1 and 2, both of them have the same structure, but they differ on the peak of absorbance of the chlorophyll, the pair of chlorophyll A. The photosystem 2, the peak of absorbance of chlorophyll A is 680 nanometer, and the photosystem 1 is 700 nanometer. So this for photosystem 2 is called photosystem 2 P680, and photosystem 1 is called photosystem P700. It's almost the same anatomy, equal. Look at them, they are the same. They differ only by this pair of chlorophyll over here. Look at my little hand. Also, so they are, imagine them. So this is my thylakoid membrane, by the way, guys. This is my thylakoid membrane, and inside of here, this is the lumen of the thylakoid. This is the space of the thylakoid space. Here is the stroma of the chloroplast. All right, that's the stroma of the chloroplast. That's the thylakoid membrane, and that's the thylakoid space. Okay, be careful. Thylakoid space, thylakoid membrane, stroma of the chloroplast. Stroma of the chloroplast. Okay, so when the sunlight is hitting on the photosystem 1 or photosystem 2, all right, those accessory pigments are going to harvest. They are going to go from staying less excited to higher excited, and they are going to move. Okay, I am more electronegative, give me your electrons. I am more electronic, give me, until they end up on the godfather for those electrons. They are there of chlorophyll A. So chlorophyll A got too much in both photosystem, too much excited. So he also go from a stage that is less excited to higher excited. And where they are going to move? They have in the photosystem 1 and 2, they have a primary electron acceptor, those orange over here. This orange over here. This is a primary electron acceptor. You will accept those electrons. Oh my God. So this is exactly what we talk here. So now I am at the primary acceptor, this one, this orange part over here, that I call primary electron acceptor. Here he is. This is where I am. This is my photosystem 2. This is my photosystem 1. And both my electrons are here now. They are at this. It's one of them is a lady, and the other one for photo is a gentleman. This is the primary acceptor of electrons. No one wants to carry that. They have to give it away. This is too much energy they are carrying. It's too much. He's too much excited. So they are going. Here's the things. They are going to give this energy is going very smoothly to an ETC, electron transporter chain. I don't want to name it. This is just call it an electron transporter chain. And they are going to move very smoothly until they end up on the photosystem 1. Because here's the thing, what's going to happen. Here is one problem. You remember, I go back over here. When the light hits, what's going to happen to my pair of chlorophyll A over here? The T-zine, what's going to happen to my photosystem 2 and my photosystem 1? They lose electron. To cover for the loss of electron, the photosystem 2 will accept, therefore, to cover electron coming from split in the water. The split in the water, hydrolyzing the water, it's under certain enzyme. I'm not going to enter to that. But splitting the water, I mean, hydrolyzing the water into oxygen and proton. So those electrons that come in from H2O going to oxygen plus proton. I'm going to balance, so half. Those electrons are going to go here to cover this loss. All right, cool. So now I'm going back to my little workout over here. So to cover this loss of electrons that's gone over here, and this loss of electron that's gone here, this one will accept electrons coming from splitting the water. What about this guy? Where he got his electron from? Because he also, in the same time when this one lost electrons from one stage, from down to up, it happened at the same time. This one, to cover this loss, he will accept electrons coming from the water. He doesn't have no sensitivity for the electrons coming from the water, the photosystem 1. He will accept those electrons, right, from the ETC, from the electron transporter chain. Isn't that pretty? It's beautiful. It's too much information. Listen very carefully. Reply it again, and you will get it. The flow of this electrons is non-cyclic. It's almost like linear. So here it is. So this is the stroma, right? That's the stroma of the chloroplast, remember? So this is my thylakoid membrane, and this is my lumen thylakoid. Okay? So this is my lumen on this, the anterior space that is in thylakoid. Okay, so if I go, I will show it to you in a picture. You remember? That's my thylakoid. That's the thylakoid membrane, which is this guy over here. And inside that is completely green, lighter green, that's the thylakoid space. And here it's the stroma. That's the stroma. That's the stroma. And that's the chloroplast envelope. Okay? So I come back. So now when I'm looking at this slide, I'm going to define where I am first. Well, okay. Well, Dr. Carbo just explained it to me. Here is my stroma. I have to understand where I am. And this is the thylakoid membrane, and this is the thylakoid space. Thylakoid lumen or space. And this is thylakoid membrane. All right. Now I identified my place. All right, let's see what's going on here. I have light. Who is hitting my thylakoid membrane? Oh, it's hitting the plant. Where in the plant? Mesophyll. Where in the mesophyll? The chloroplast. Where exactly in the chloroplast? thylakoid membrane. What we have in the thylakoid membrane, we have two photosystem, 1 and 2. Okay. And those photosystem are going, what photosystem 2 is very sensitive to accept electrons coming from the water. When the electrons pass from less energy to higher energy, and this one also, he go from less to higher energy. This one, to cover the loss of electron, he will accept the electron coming from this plastic, it's ATC, it's an electron transporter shell, okay? Right? And this one, he will accept it from the split in the water. So that's my ATC, okay? It's a BF complex. All right, those electrons that get excited in photosystem 2, they go through the ATC. What happened to the electrons that get excited in the photosystem 1? Well, this is where they give it to another carrier of electrons. And this carrier of electron is almost the same that the carrier of electron that exists in the mitochondria. It's an NADPH. It's an NADPH. Remember, that's the only one that carry and store this electronic energy. Store it. It gets reduced to NADPH. And also, he doesn't even give it to him directly. He gives it to him through an ferrodoxin, which is also another molecule, very not complex. Okay, so he formed NADPH. NADPH is also another source of electronic energy. Remember what I told you before. When an electron travels, to not create potential actions, a proton travel. So the electrons, the one that I am mentioning here in black that you are seeing are the electrons. They are going, and there they end up in a. What's happened to the proton? Where do you go from there? Going up, going down. Anytime a proton is going, anytime the electrons are going into the other molecule, protons are also going, because the water is splitting also in proton. So I end up having this side of the membrane is in contact of, but remember that this is my thylakoid human for space. Very positive. Very, very positive. Because of the presence of the accumulation of the proton. So we end up having this side of the membrane, because it's a bilayer membrane, thylakoid more positive than this side of the membrane that is in contact with the stroma. Thanks God. I have also an ATP synthase, where the rotor now, the rotor of this ATP synthase is in contact with the stroma. So therefore, those proton, remember, this membrane is a bilayer of phospholipid. Even single molecule of proton ion can cross this membrane. You need a transporter. The only transporter of proton is this ATP synthase. Okay, so he's going to cross this ATP synthase to go diffuse into the stroma. To not accumulate too much positivity over here in this part of the membrane, too much positivity. So they have to go out and therefore diffuse in the stroma, not staying in contact of my membrane, to not create potential actions. And in the same time, what I done, I establish my potential action, my resting potential action, which is almost around, it depends on the cells, between minus 70 millivolt to minus 120 millivolt for some some cells. Anyway, so I cross it, right? And what I told you about ATP synthase is very sensitive when a proton cross it. They get stimulated, and when it gets stimulated, it's ATP synthase. This enzyme is going to phosphorylate ADP to ATP. So therefore, I produce ATP. Look at that. Isn't that pretty? I produce ATP but a system that we call photophosphorylation. Photo means light, is by light. The only thing that stimulates this production is the light. So I call it photophosphorylation, photo for photon, which are the particles of the light. So therefore, what's happened during my light reactions on cyclic electrons flow, because it's linear, it's not cyclic. It's going from it's one way, right? This is, I produce ATP, I produce NADH, it's a lot of things that happen. Summarize, I produce ATP, I produce NADPH, all right? And they produce oxygen, they split. So what I start here with, I start with the sunlight, with water, they have the two photosynthesis, and they have NADP plus, and they have ADP. This is what I start for. And they produce oxygen. Oxygen, if you remember the equations, is one of the, if we remember the equation for photosynthesis, all right? Remember I told you to make a paper? It's one of the products. So remember, when you have to go see your aunt that is in Fort Worth, and you have a friend in Arlington, so when you are going from Dallas to Fort Worth, you traverse Arlington. It's in your way. So your friend will tell you, or you tell your friend, okay, I am going to stop by. You didn't reach your final destination. You are stopping by. So this is what's happened. Did I finish my photosynthesis? Did I have my organic molecule? Not yet. All what I done is splitting my water. So I produce actually oxygen as byproduct. So this is what I done. I produce oxygen by byproduct. I produce ATP and they produce NADPH. There are some systems that they are not linear. This is a cyclic electron flow. Only ATP is made. There are no NADPH. We have only one photosystem, which is the photosystem. They are photosystem 1. And this is to conserve their energy. So this is the photosystem 1. So always they are doing the electron go from one state, go to the electron transporter and be recycled. And be recycled. And during the process, ATP is formed, almost the same process that has been formed during the non-cyclic light reactions. I have here some videos I want you to watch. Now we are going to pass to the carbon cycle. Carbon cycle is a cycle that will start with one molecule and it will produce actually my organic molecule, and this it will be recycled. And what is going to be recycled is a molecule that will be in this. Remember, the source of carbon here came from CO2, and CO2 is a gas. So first of all, I have to fix it into an organic molecule, which is very unstable. So is exactly what's going to happen during my carbon fixation or cycle. You remember those ATPs and NADPH that has been produced during the light reactions, where they are going to be used as a source of energy to produce this. Remember, this is an anabolic energy. The cell, the currency that the cell will use to fix the CO2 have to be ATP, have to be an electronic energy into the NADPH, because this needs a lot of electrons. This is the acceptor of electrons over here that they are coming actually from the splitting of water and stored into the NADPH. So we need 12 molecules of NADPH, 18 molecules of ATP, and 6 CO2 to produce 1 molecule of carbon, glucose, C6H12O6. And remember that I produce also water 6, but I'm using 12 molecules of water, so I'm not going to talk about this 6 molecule of water that has been produced in the end, because I'm going to talk only about using 6 molecule of water. Okay, so the structure of the carbon cycles is very easy. First of all, I have to fix my CO2. And then I pass to a stage that I am going to reduce the CO2, and then I pass to the stage that I am going to regenerate the acceptor of CO2. This is catalyzed by an enzyme that we call Ribisco. Ribisco biphosphate is a molecule of carbons that is biphosphate, is the one that will integrate the CO2 and the Ribisco enzyme into a 6 molecule of phosphoglyceraldehyde triphosphate, PGA. And this 3-PGA, I'm going to stage of reductions, and here I'm going to use you remember the ATP that has been formed during the linear reactions? The right reactions, I'm going to use it. And also I'm going to use those in electronic energy coming from NADPH to produce glyceraldehyde triphosphate, which is the simplest molecule. This is the simplest. So 2 of them came together to make a molecule of sugar, because glyceraldehyde triphosphate, it will combine with another glyceraldehyde triphosphate and they can make C6H12. This is 3 carbons molecules. I make 6 of them. So anytime, first of all, 3 molecules of CO2, look at that, because I have 6 molecules of CO2 in the beginning, all right? 3 molecules of CO2 will produce a molecule of glyceraldehyde triphosphate. 1 of them will come out, okay, I am out. I don't make my molecule of glucose yet. And then the rest are going to regenerate the Ribisco biphosphate, because this is a 5 carbons molecules. And the Ribisco will give me the 6 unstable molecule that will be reduced to produce this 3 carbon molecules. The 3 other molecules, the CO2, will do the same thing. It will enter the Calvin cycle to produce another molecule of glyceraldehyde triphosphate. 2 of them came together to make CO2. So in the end, CO2 will make 1 molecule of C6H12O6. And during this, so the Calvin cycle, the reactant for it are going to be CO2, ATPs, and NADPH. CO2. This is what I start with. And then we need what? We need a fixator for CO2, which is the ribiscose biphosphite, which is 5 carbons, and the enzyme ribisco to produce 6 molecules of 3-phosphoglyceraldehyde. So by 3 CO2, I am using 9 ATPs. For 3 CO2s, 9 ATPs. For 6 CO2, I'm using 18 ATPs. Everything's have to be double. Okay, I want you to look at this. You will get it. I have here a very nice video for the summary photosynthesis that I want you, and they have here a practice. So photosynthesis is an anabolic reaction. So as per this comparing, I want you to do your table yourself and come see me or contact me if you need more information. Let's end up our lecture for today. All right. Any questions?